guys, BIOBEAN here. So in our last video, we talked about meiosis. So in this video, we're going to talk about what happens when meiosis goes wrong. So let's get started. So when meiosis goes wrong, we call this non-disjunction. During anaphase of meiosis 1 and meiosis 2, the chromosomes are being pulled apart to the different ends of the cells. In the case of meiosis 1, the whole chromosome pair is being pulled to either end of the cell, whereas in meiosis 2, the sister chromatids are being pulled apart into either end of the cell. So when this goes wrong, it is called non-disjunction. So this is when the chromosomes or chromatids are not properly pulled to either end of the cell. So in the case of meiosis 2, it would be both pairs of chromosomes getting pulled to one side, which would result in one set of cells having both chromosomes and one cell with no chromosomes. And then if that occurs in meiosis 2, it would result in one cell containing two chromatids and one cell without any chromatids. So let's get a little bit into what that would look like at the different stages of meiosis. All right, so first we're going to start with non-disjunction that occurs in meiosis 1. It's very important that you know how to follow through the chromosomes if non-disjunction occurs, because they love to give you questions asking about if there was non-disjunction here, what would be the result, and questions like that. So it's important that you're able to follow this through. So here we have metaphase 1, where our tetrad or homologous chromosome pair is lined up along the metaphase plate. So if non-disjunction happens during anaphase, that means that both chromosome pairs are going to be pulled to one end. So if non-disjunction happens at anaphase 1, those chromosomes are going to be pulled to one end of the cell, and then during telophase, it is going to split those off. So the chromosomes that were on one end of the cell are going to end up in that one cell, and it's going to have both pairs of homologous chromosomes, and then one cell is going to have no pairs of homologous chromosomes. So now let's follow this through to see what the gametes would be made at the end. So if this cell now contains two pairs of homologous chromosomes, and we go into metaphase 2, they are going to now line up single file, because in metaphase 2, they no longer arrange as chromosome pairs. Even though they're the same chromosome, they're going to line up at different spots within that metaphase plate and line up in a single file line. So now, following that non-disjunction through that occurred in anaphase 1, we look now and see during metaphase 2, the two are going to line up independently along that metaphase plate, and the cell that received no chromosomes is going to continue to have no chromosomes. And so then when anaphase 2 occurs, the chromosomes in the cell that contains chromosomes are going to pull them to either end of the cell. So remember, when we say non-disjunction in meiosis 1, it is only a single non-disjunction event that occurs in meiosis 1. So there isn't another non-disjunction event. There's only a single non-disjunction event. So now that we're in anaphase 2, they're going to segregate properly. So they're going to go into the opposite ends of the cell. So now let's see what would happen at the end of telophase. What are those gametes that are made if non-disjunction occurs in anaphase 1 of meiosis 1? So now, following this non-disjunction at meiosis 1 through, we see that we create 4 gametes. So the 2 gametes that were created from the cell that received no chromosomes is going to contain no chromosomes there. And the cell that came from the cell that got an extra set of chromosomes, they're going to carry 2 copies of each chromosome. So now they're going to be 2N for that particular chromosome, and this is going to be 0N for that particular chromosome. So you have to remember, when we're looking at diagrams like these are simplified diagrams. So I've simplified it to only show you a single chromosome pair. And we're looking at non-disjunction that occurs within that single chromosome. We have to remember that humans contain 23 chromosomes. And so while there was non-disjunction that occurred within this chromosome, which we always call chromosome 1 just for ease of it, the other 22 chromosomes are properly segregating. So while I say that these cells have no chromosomes, it is only no chromosomes in reference to the chromosome that went through non-disjunction. So if the rest of the cell that has no chromosomes is really going to contain 22 other chromosomes that were properly segregated into all these cells. So when we say no chromosomes, we are talking in reference to the chromosome that had non-disjunction occur. Alright, so now we're going to follow through what happens if non-disjunction occurs during meiosis 2. So it's going to be happening during anaphase 2 of meiosis 2. So something I just want to mention here, you don't see prophase in this diagram because I cut it out just for ease of explaining it, and so it all fit on the board nicely. So I've already drawn out meiosis up into metaphase 2, because if non-disjunction is occurring in meiosis 2, that means that the previous portions of meiosis are happening totally normally. So during metaphase 1, the tetrad or homologous chromosome pairs line up along that metaphase plate. During anaphase 1, they get pulled to either end of the chromosome, and then telophase 1 separates those into their individual cells. So now each cell is going to contain a homologous chromosome. And so now we're in metaphase 2, and they have lined up along that metaphase plate. And remember, in metaphase 2, they're lining up in a single file line. So now we have to follow through non-disjunction. And so we're going to have to pick which cell is going to have non-disjunction. Because as opposed to non-disjunction that occurs in meiosis 1, we're only dealing with a single cell. But now we have two cells, and just because non-disjunction happens in one of the cells doesn't mean it's going to happen in the other, because they're totally independent cells. And non-disjunction is a mistake that happens, and it's not something that happens very frequently. So I'm going to arbitrarily pick which cell I'm going to follow non-disjunction through in. So I just arbitrarily chose this right cell to be the one that non-disjunction occurs in. So if this non-disjunction only occurs in this cell, that means that the meiosis 2 in this cell occurs completely normally. So we know that when non-disjunction in meiosis 2 happens, it is going to create two cells that are completely normal because they are completely unrelated to the cell that is going to have non-disjunction. So if we have non-disjunction in meiosis 2, that means during anaphase 2, both sister chromatids are going to be pulled towards the same end of the cell. So instead of them being separated, they're going to be pulled together. And so now, let's see, what does that result in telophase 2? So now, looking at telophase 2, we can see that it results in one cell that is going to have no chromosome, and one cell that has both sets of sister chromatids. So they kind of look as separate sister chromatids, because while I showed the full X kind of pulling apart, it's really the two individual sister chromatids that are going to be pulled apart. So you see, those are the two sister chromatids that should have been one in each cell, but because that non-disjunction happened in anaphase 2, it is going to result in one cell with two chromosomes, so both of those sister chromatids, and one cell that has no chromosomes. So again, remember, when I say no chromosomes, I'm speaking only in reference to the chromosome that had non-disjunction occur, whereas the other 22 chromosomes are going to separate properly into all of these cells. So at the end of non-disjunction in meiosis 2, we end up with two cells that are completely normal, one cell that has two copies of that chromosome, both sister chromatids, and then one cell that has no copies of that particular chromosome. So when this non-disjunction event happens and we follow it through to the end of telophase 2, we see that there are differences in the chromosome copies across all of the gametes that are made. And we call this aneuploidy. So this is when they have a different ploidy than what you would expect. And so ploidy refers to the coefficient of n. So normally we would expect a human cell to be diploid, but in the case of non-disjunction, it can cause there to either be more chromosomes than there should be or less chromosomes than there should be. So we call this aneuploidy. So the way that I kind of try to remember this is I break up the word into aneuploidy. So I call it aneuploidy that comes about as a result of non-disjunction. You get a new type of ploidy that you would have not seen in a normal cell, whether that being more chromosomes that you would expect or less chromosomes than you would expect. So there are different types of aneuploidy depending on which gamete gets fertilized. So one of the types of aneuploidy is trisomy. So that means you have 3 copies of one particular chromosome. So this would happen if non-disjunction occurs in one of the parental cells, resulting in one gamete that contains 2 copies of the same chromosome. So that means we started with one gamete that was at 2N, and that met another gamete that was only 1N. So then when we have a 2N and a 1N come together, that is going to create a 3N cell in respect to that particular chromosome. So remember, when I'm talking about 3N or 2N, I'm only speaking about the chromosome that had non-disjunction occur within it. And so we rate trisomy based on which chromosome had the non-disjunction. So we're going to go over a few types of trisomy based on the chromosome. So we classify trisomy based on the chromosome that has the trisomy or the non-disjunction occurred in. So trisomy 21, probably the most well-known form of trisomy, causes Down syndrome. The next one we'd like to look at is trisomy 13, and this leads to Patau syndrome. And trisomy 18 leads to Edwards syndrome. So all of these lead to large defects within the infant that's being born, and in some cases, and particularly with Patau and Edwards syndrome, lead to death very early on. And so this tells you how important it is that these chromosomes segregate properly and everything occurs in the correct way. So here we can see trisomy 21, 13, 18. These are all non-disjunction events that occur within the normal somatic chromosomes. So let's look at what happens if there is non-disjunction in the sex chromosomes of an individual. All right, so now we're going to focus on what happens if non-disjunction occurs in a female with their sex chromosomes and if it occurs in meiosis 1. So we're going to see the resulting effect once it hits a sperm. So let's follow it through the non-disjunction. So we have the homologous chromosome pairs line up along that metaphase plate, and then anaphase is going to occur, and this is where our non-disjunction is going to happen. So both pairs of homologous chromosomes are going to go to one cell, and then the other cell is going to have no pairs of that homologous chromosome. And so let's see what the resulting gametes are. So here we have two gametes that are not going to have any X chromosome, and two gametes that are going to have two sets of X chromosomes. So I kind of made an X with a red because I want to show you that this is an X chromosome that we're talking about, but I want you to keep in mind that they're not actually going to be an X at this point. They're going to be the individual pairs of chromatids. So the red X is just to denote to you that this is the X chromosome where the non-disjunction happened. So we're going to start with the example of this sperm containing an X sex cell. So all men have an X and a Y chromosome, and so they are either going to make a female if they donate an X, or they're going to make a male if they donate their Y. So in this particular case, this sperm is donating their X chromosome. And so if it meets this cell that contains no X chromosome, so this cell or this cell, you are going to get Turner syndrome. So when someone only has a single X chromosome and no other sex chromosome, that is going to be called Turner syndrome. So what happens if this sperm containing our X meets these cells that have two sets of X chromosomes? Well, then that is going to cause triple X syndrome. So they're going to have 3 copies of an X, and that's called triple X syndrome. So what happens if that sperm is carrying a Y chromosome? So if that sperm is carrying a Y chromosome and it hits one of these female gametes that doesn't have any sex chromosome, that is going to be a non-viable zygote. So if this Y hits one of these that doesn't have any other sex chromosome, that baby is not going to become fully developed. It is going to miscarry before that occurs, or maybe even implantation might not even take place. So what happens if this sperm carrying the Y chromosome meets these female eggs that are carrying both 2 copies of that X chromosome? So I just want to remind you, here's a little aside, I've made this Y chromosome with a piece in red, just to remind you that it's sitting as a single chromatid, right? This is at a 1N and 1C value. So I show you the X and the Y merely just so you know whether we're talking about the male or female sex chromosome, but they're not actually sitting there as an X or a Y, they're sitting as a single chromatid. So if this male Y chromosome hits, or rather the sperm carrying the male chromosome hits one of these female chromosomes, or rather these female cells that contain the 2 X chromosomes, that is going to result in Klinefelter syndrome. So when you have a XXY, that is going to lead to Klinefelter syndrome. So that concludes our video on non-disjunction. Stay tuned for our next video on the different life cycles of the kingdoms of life. And please don't forget to comment, like, and subscribe in order to continue to receive updates as these videos come out.